Okay, and I'll go to the next part about my scientific talk that I would want to share with everyone for now. Actually, I want to discuss and one of my interesting and also one of my research that really went about this topic. I'll talk about the hyperoxia in CPB and ECMO. Okay. I chose this topic because this is just published in April this year in American Heart Association Journal in the heart failure section. And they show interesting data for me. The number from the also registered and the patient is almost 10,000, 9,959. That's almost 10,000. That's a huge number. Actually, it's a retrospective study, but it's a huge number, and the data very clear to show us for something. The title of the paper is Exposure to Arterial Hyperoxia during Extracorporeal Membrane Oxygenator Support and Mortality in Patients with Cardiogenic Shock. They look at the data and divided in 3 groups. Look at the PaO2 with defined for the hyperoxia. The normoxia, the definition is 60 to 150, and then compare with my hyperoxia 151 to 300, and compare divided into group. And the last group is called definition is severe hyperoxia, which is more than 300 PA for the millimeter mercury for PaO2. So that means we have 3 groups to compare: normoxia, mild hyperoxia, and severe hyperoxia. And then they show the mortality that's happened. It's quite surprising for me. I mean, I'm so very interesting. The PaO2 measure after 24 hours of ECMO. For me, 24 hours is not too long, because the ECMO sometimes 7 days. It's long for ECMO. So when we measure at 24 hours, for me, maybe I'm wrong, but I just want to share and discuss with you. I think 24 hours is not too long, but the data show and very clear show the normoxia compared with mild hyperoxia and severe hyperoxia. The mortality rate is higher in severe hyperoxia group. And also, yes, of course, that's a different from statistic. And the statistic is very, very strong to show the different, the phenomenon happen. And if you compare between mild hyperoxia and normoxia, yes, the mild hyperoxia, even 151 to 300, the mortality rate is significantly different from normoxia. Also very interesting from this data. Before I go to next slide, I want to talk for this slide a little bit. In 2019, the Nobel Prize Award for the group of scientists, the three scientists, which discover about how cells sense and adapt to oxygen in condition of low oxygen, that's we call the hypoxia. And data that's very, they show the pathway. In normoxia, when we have a sufficient oxygen, the HIP-1 alpha was degrade. You see this pathway. In the normoxia, we have the sufficient oxygen. The HIP-1 alpha finally is what degrade. Nothing happened in the cell. That's normal happen. But in hypoxia condition, because you don't have enough for oxygen, the HIP-1 alpha is stimulated at the DNA level and affect on the transcription factors. Let's think about coronary heart disease. We know that we have the collateral vessel. The question is how collateral vessel happen, how angiogenesis happen at that time. That's because as a hypoxia happen, and then they switch to this phase, and then finally they send and they behave. So many things happen they change the body function. So many things. And let's think about the cancer. That's why the same pathway that's happened, and they have and finally have the angiogenesis happen in cancer cell. from this discover that we can think we can like develop medication like to if we can stop the HIF1 alpha, that we can stop the cancer cell. Actually, there's a huge impact and very clear data to show hypoxia, how cells send hypoxia. Too easy word to say, I would say we have the oxygen sensor in cellular level, what we call the HIF1 alpha in the hypoxia condition. As we concern, and I truly support, yes, we need to concern about hypoxia. We need to concern about the hypoxemia, anything. Yes, of course, that's we concern for the perfusionist. But in contrast, how cell sent to the condition of hyperoxia. And when I read the paper that I show you about the ECMO, the first two slides that I show you about the ECMO, and I think, what's about the hyperoxia? And at the same time, March this year, they just discover very interesting data and published in high impact factor in the cell molecular levels. And they show even the high oxygen, about 30% oxygen, but for perfusion, sometimes we open for 60, it's dependent protocol, right? But even a return to from 21 in normal, you increase to 30%. That can be oxidized one pathway. This pathway they call the iron sulfur cluster, and then that became oxidized. And finally, it's like a degraded protein, some protein in our body, and finally the cell stop function. That's they show from the data, and they use the techniques that we call the CRISPR technique, very wonderful technique to delete gene or added gene and to show how body function. And data is very nice, very, very interesting. And I'll show you three more slides before I'm ending my talk. And when we look at the physiologic level in our physiology, like a perfusionist way, I will to show for this diagram. I'll start with the oxygen content. Yes, we know oxygen content is depend on hemoglobin, is depend on oxygen saturation, and just a few effect from PaO2, right? PaO2 multiply 0.003, and then plus just a few happen. Anyway, if you look at this data, when you increase oxygen content, let's say PaO2 400, I'll go back to this again. When you increase PaO2 400, 600, that means oxygen content will increase. That's happened from the mathematical formula because you increase the multiply and then you plus. So from the DO2, the DO2 is cardiac output for perfusion, is a flow. DO2 is cardiac output multiplied by oxygen content. When you increase oxygen content, you shouldn't increase DO2, right? But when you go back, and this is happened at the coronary, at the heart, when you go back to see the data, they conclude from so many studies, they show that when you increase oxygen content, oxygen delivery at the coronary, it doesn't increase. It does not increase anymore. It shows decreasing. Why is decreased? Is this effect from cardiac output or not? If you think maybe reduce stroke volume, maybe some mechanism involved through heart rate, but so many studies show it's not effect from stroke volume and heart rate are not changed in this condition. If you increase PaO2, both of it does not different. But what they found is reduce the flow at the coronary. So that way the thinking about maybe this increased resistance, the resistance increase at coronary, maybe from vasoconstriction. That's why, like a reduce flow, finally decrease the DO2 happen. That way the question why vasoconstriction happen, why it's happen. And if you look at the brain behave the same way at the heart. You increase the PaO2, which you increase the oxygen content, but the O2 is not increased anymore. And also they show the increase the flow, which is that means the vasoconstriction happen in somewhere in the brain. But it's different from skeletal muscle. The skeletal muscle contrast from heart and brain. It's like a similar way you increase the oxygen content, that DO2 also increasing. The question is why. I try to read so many papers, and as I mentioned, I want to discuss with everyone. Maybe we don't have the exactly the clear explanation about this, but some review there. Actually, it's the review article just published in 2018 and 2022. One reason that interesting that may be hyperoxia in the radical oxygen species, right? And then finally being create the oxidative state, and that's make the nitric oxide imbalance. So that's mean red vasodilator, that's may cause vasoconstriction. Yes, that's possible. I think I do agree with this. And also in animal study, they show the T at the high PaO2 greater than 300 millimeter mercury in its effect on increasing the inflammatory cytokine like the TNF alpha in the interleukin 6. And the last one that they show in hyperoxia condition, the plasma ATP concentration were changed. That may affect on the vasodilator signaling pathway. And now I'll show you just a few more slides from my research. From the last reason that I found, maybe reasonable in somewhere, in some point, I think ATP have two receptors, P2X and P2Y. And from my research during my postdoc time, when you increase renal arterial pressure, we found that it increased interstitial ATP, and interstitial ATP bind with the receptor, P2X receptor, and finally that's called vasoconstriction. Then I show you at renal perfusion pressure, that's from my lab, renal perfusion pressure at 100 that show the renal afferent arterial around here, that's quite big. When you increase to 140, it shows smaller, which is the renal autoregulatory happen, and also effect from ATP by with P2X. And then when we block P2X receptor, and it turned back to normal value, which is clearly show the effect from ATP by with the receptor. That's more reasonable if hyperoxia induced to increase ATP in some point. That's may cause vasoconstriction. At least at kidney, that's happened from my research. But the question is, what's about the dose and time to exposure to PaO2? For example, yeah, the data show from ECMO, but ECMO, so 24 hours. Let's think about the CPB. Sometimes CPB 1 hour, 2 hours, and we maintain. It's, I think, depend on protocol of the center. But like I share with you, for me, I do about 250 millimeter mercury that I control PaO2. Is the time and the dose effect on the harmful or not? The clinical trial just show in 2019, they compare into group. The group one, they maintain the PaO2 allow 400 to 500 during the CPB. And group 2, they control the PaO2 about 100 to 200, it's around here. And then they compare, they look at the hospital mortality, stroke, cardiac arrest, acute kidney injury, and mesenteric ischemia. And they compare and they look at the data. And they summarize that hyperoxia during cardiopulmonary bypass does not decrease cardiovascular complication following cardiac surgery. On another word, that means hyperoxia or normoxia. It does not different. It's the same way. It's the same happen for cardiovascular complication that they look at. So we may think that for short-term exposure, for example, like CPB, maybe hyperoxia may not be harmful, but ECMO we may need to think about, right? Because the data quite clear to show us. Actually, I have very short conclusion about this. That's from my heart and from my idea. I think too much or too late oxygen is toxic. But we want to know, and what exactly number, but we don't have the answer for now. I mean, we need clinical trial, we need observation research, so many things, but I mean, we keep in mind maybe too much or too late. Yes, he's toxic. And I will end my talk as a perfusionist. I love discuss in my personal a lot. I share with my student, perfusionist is hardworking 1,000%. You need to some skill, the extra skill about 500%. And you sleep late, you sleep just only 0%. You work all the time for the emergency. And you need coffee, so maybe more than 100%. That I talk with them, and my students say, and you back pain 500%. That my students said, oh yeah, I agree. As I said, I would like to end up my talk with to acknowledge with some people that incredible people that I met. First, I'll contribute my talk. If a good thing happened for today, that's my talk. I'll contribute all of the good thing to Dr. Gabriel Navarre. I learned a lot from him. Not only physiology, but scientists, the opportunities. I can't believe that he accepted me when I wrote the long email to him, and I told him I am a perfusionist. I can't believe that you accept me. I'm very grateful, Dr. Navarre. I'm not sure that you attend my talk or not, but if you attend, I'll say hello to you and say hello to New Orleans. Yes, and thank you for Joe, and also thank you for the director of the Texan Heart and the president of the APCP that I met. Yes, finally, Thailand, Bangkok is too far from Houston, but we different from night time and daytime, but we are being here because our love in perfusion bring us together. So thank you so much. Thank you.